ELLIE MCNUTT, thanks so much for coming on to Evolution Soup all the way from your office in Ohio. You are an assistant professor working in the Department of Biomedical Sciences at Ohio University Heritage College of Osteopathic Medicine, and your main interest is in human evolution and how we came to walk upright. We've talked about human bipedality before on this channel with Matt O'Neill, as well as your colleague and co-author, Jeremy Da Silva. But in this interview, we'll be walking in the footsteps of the most famous hominid of all, 3 million-year-old Lucy. So how are you doing, Ellie? Your recent paper is all about footprints attributed to ancient hominids that were contemporaries of Lucy and her species. So what has the response been to this work since its publication?
I'm doing really good. The response to this has been really fabulous. So this paper is about footprints, and footprints, I think, uniquely in our fossil record, capture people's imagination. And so a lot of people have been really keen because they're kind of a cool snapshot of a moment that's very ephemeral, that we don't kind of think about, whereas bones are neat, but they're sort of, okay, I grabbed a bone out of the ground, and this has been really cool. So it's gotten a lot of attention and sort of captured a lot of people's imagination, both folks just out in the world and then also within our scientific community, and sort of sparked some really cool conversations as well.
Well, before we start our walk with Lucy, let's just hear a little bit about your background. Ellie, what was the path that led you to where you are now, studying human evolution?
So yeah, my path is actually quite circuitous, which I think is a pretty common thing sometimes for science and how we end up getting to places we didn't mean to. So I grew up, originally I'm from Iowa, so grew up on a farm. And my mom actually has her PhD in microbiology. So when I was quite little, I kind of assumed everyone's mom had a lab and was quite surprised when I went out into the world and discovered that that wasn't true. So I was always around science and really interested in science and kind of how we interact with the world. And so I actually started my sort of college career as an engineer. I was really interested in materials engineering, which is less how do you build a bridge and more what do you build a bridge out of. And I was really interested in applying that towards like prosthetics. So instead of using titanium to fix a shattered ankle, you'd use a ceramic that would essentially trick your body into thinking it was bone and would get grown back into it. Yeah. And I sort of was thinking medical applications and things like that. And I started working in a lab that studied muscular dystrophy. And I kind of had a mind-blowing moment of like, squishy science is awesome. What am I doing? Because I sort of realized that while I enjoyed being an engineer and there were parts of it I loved, and I was a good engineer, it wasn't what made me happy and passionate. I wasn't keen to be doing that for the rest of my life. So I sort of switched at Iowa State, which is where I was doing my undergrad, into biochemistry, which is the closest thing we had there to physiology. So I was going down that path instead, and I was taking anthropology classes for fun, just because I like it, and it was cool, and I liked the skeletons, I liked the history, and I got really lucky, sort of in the right place at the right time in one of those anthropology classes, and my professor put on this documentary about Australopithecus, and this guy walks onto the screen, and he's got a tibia, so a shin bone from a modern human, from a chimpanzee, and from Lucy, who we'll talk about more today. And if you look at the bottom of the shin bone, it's fairly square in a human because we put the forces through our ankle pretty uniformly. They kind of go all around. Whereas if you look at something like chimpanzee, they're trapezoid. They're wider at the front than they are at the back because when they climb, they end up putting a lot more of their weight through the front than they do through the back. And so if you look at Lucy, she's 3.2 million years old. She looks like us. And it shows that she was moving in some of the ways that we do. And so it kind of blew my mind because it was sort of the meeting moment of biochemistry, about how bones form and how they respond to loading, biomechanics for how do you understand the way that living organisms move. And sort of that was the gateway drug into being a biological anthropologist. And I sort of said, I want to do that. How do I do that? And I ended up applying to work with the person who was in that documentary who ended up becoming my advisor. And so I did a master's in biological anthropology at Boston University and then completed my PhD at Dartmouth College and then went to a postdoc working at Keck School of Medicine at USC out in LA, because when you're working with human fossils, you only have pieces of the puzzle, right? You end up with a knee here and a bit of a shoulder and things like that. You have to kind of find a way to click those back together to make sense of fossils in the past. And so one of the ways you do that is you have to be pretty good at human anatomy to kind of understand how these disparate pieces come and click back together. So now I do a lot of work teaching anatomy, mostly in med schools, as sort of my day job, that then significantly overlaps with what I do in terms of my research. And so now I serve as an assistant professor teaching anatomy at the Ohio University Heritage College of Osteopathic Medicine. So that's sort of the long pathway around engineering all the way over and back around to now anatomy and continuing this sort of research into why we move the way we do, which is kind of the basics of what got me interested in engineering in the first place. So that's the plan.
Lucy was discovered in 1974, a fantastic example of an upright walking hominid from deep in our past. The reasons for bipedalism, walking upright, is in many ways the holy grail of paleoanthropology. So what does your own research involve, and why is it important for the understanding of this subject?
Yeah, so sort of the titles of the hats that I wear involved in my research. I am both a functional morphologist and a comparative anatomist. And so functional morphology is essentially trying to make a connection and understanding that the fact that what we do and how we move through the world changes and shapes our skeleton. So for an arboreal animal that climbs all the time, that's going to change the way our skeleton is designed and how we can actually see those bones shaped, even though largely you could pick up a femur from me and a femur from a bear and a femur from, a giraffe, and they all look like femurs, but they aren't the same. And those differences between them are meaningful, and they tell you something about how that animal is moving through the world. And then comparative anatomy. So obviously the work that I do, I'm trying to understand the evolution of human locomotion. And it would be fabulous if I had a time machine and I could just go back 6 million years, 18 million years, and look at some of the primates that have been along our lineage and actually figure out sort of, what were you doing? Why did you move this way? What was going on that maybe, predisposed us to stand up? But obviously we can't do that. So the best way we can interpret these parts of our history are to find the fossils of those animals, take them out of the ground, and then we have to compare them to something. And obviously the only name in town in terms of humans is us. So humans become one of the best examples of sort of, what do our own bones look like? How do they change if you have a group that climbs a little more or less or does other things like that? But then obviously we need a broader spectrum than that in order to actually really understand things that are going on in our fossil record. And so that means looking at our close living relatives. So these are things like chimpanzees, gorillas, the other living primates. And it means looking at things that are distantly related to us. So a lot of the work that I do involves looking at bears, because bears are a really good analogy with the living apes. They're large-bodied, they climb a lot, and they have plantigrade feet. And so plantigrade is what I'm really interested in. It's kind of the fancy word for heel striking. So when humans hit the ground, our heels hit first, and then the rest of our foot goes as we walk. So we sort of heel strike, and then we end up pushing off with our toe. And that's a plantigrade animal, as opposed to something like a cat or a dog, which would be digitigrade. They're up on their toes, and their heels never make contact with the ground. And that's really important if you want to understand bipedalism, because in order to stand up and be a good stable biped, walking around on two legs, you have to have a nice plantigrade foot. Which is why if you ask your cat or your dog to stand up on two legs, they can do it, but they're wobbly, they're uncomfortable, and you're really only going to do that if you've incentivized them through treats or something, or if you have an animal.
Because our feet are more flat, isn't that right?
Our feet are more flat. They have nice arches, which are going to help us with the energy of that, but they're meant to hit the ground and have the whole of the foot press across the ground. Whereas some of these other animals are either touching, like a horse is truly up on the tiptoe, and then a lot of things like cats and dogs are up on the tips of their fingers, but not on and toes, but not on the whole of the foot. So their heels would never touch the ground. They're always going to be up in the air, which makes them nice, fast animals, but they're only really stable when they're on all fours. Right. And so that's what I kind of do. A lot of folks certainly of my dissertation research and what I'm kind of coming out of now is if you can figure out what primates precede the hominin lineage. So Hominins are anything on this sort of line coming down to us. So these are our human ancestors and our cousins. And we want to figure out the circumstances in which bipedalism happened, because this is kind of the thing that makes us so different and unique from a lot of other animals on the planet. So really, in terms of mammals, the only other animal that is upright on two legs all the time is a kangaroo. Obviously moving through the world a very different way than we do. And then outside of mammals, things like birds, but that's really kind of only when they're on the ground. Most of the time, most of them fly. And so this is a pretty strange way of moving. It's kind of definitely one of the earliest defining characteristics of our family lineage. And so that's why it's kind of one of the holy grails of our science to figure out why did this happen and kind of what drove that. And one of the ways to do that is to sort of narrow down the field of possibilities of animals that may have been, sort of ancestors to us. And so the work that I do is trying to say, all right, I know what the feet of an animal who are plantigrade look like by using this comparative anatomy, functional morphology. Can I then apply that understanding to fossils of primates, sort of apes that lived anywhere from 20 million years ago up to about 5, and try and narrow down the field of like, these folks have feet more like us as opposed to something like the living monkeys today who are semi-digitigrade. They walk on their toes, but their heels are well raised. And say, all right, these animals are good candidates. Where are they? Are they hanging out in trees? Are they hanging out on the ground? Like, try and make that big mystery a little bit smaller and help us kind of narrow in our focus and kind of figure out where bipedalism came from then. So that's sort of the basis of what I'm interested in, as well as within our own lineage, trying to put together, if you find a hominid, how are they moving? Are they moving differently from one another? Kind of how did that change even within our own lineage? Because certainly we have not moved exactly the same way for the last 6 million years since we split from chimps. And so figuring out all the little evolutionary experiments that happened throughout our lineage is also what's really cool and interesting to me.
Let's just focus on Lucy and why she's so significant. The fossil skeleton was the first example of Australopithecus afarensis ever found. Almost every hominid discovery since Lucy is compared to her in some way. So what is her story?
Lucy is fabulous. She's one of my favorite fossils ever discovered, and she's amazing. She's got a really cool story about sort of the luckiness of finding a fossil. So Lucy is an example of Australopithecus afarensis. She's about 3.2 million years old, and what makes her really fabulous is she's a really complete skeleton. So we have about 40% of her that was found. And she was originally discovered by Donald Johanson and Tom Gray. And so they were working in an area of Ethiopia called Afar. So this is the region there. And they had been to the Afar region for a couple of field seasons before, found a couple things. The year before they found a knee, and we can talk about that more in a second if you want. But Lucy, Donald Johanson had actually gone out to go look at a different locality. So fossil sites are split apart by sort of gridded out. So this is this area, and this one is 288, and this one is 333, and things like that. The number of grids to them. And so the way that we find fossils, particularly in Eastern Africa, is that they are inside essentially these hillsides, and we have to wait and see kind of at time is part of the way that we find them. So we have an opportunity, seasonal rains wash off part of a hillside and expose fossils that you wouldn't know were there before, because obviously they're stuck inside the hillside. So he'd gone out in the morning, I think it was in November in 1974, to go look at this other field site and spent the day. It was getting much, much hotter. It got to be 110. He and Tom were getting tired, and they wanted to go home back to base because it's kind of the hottest part of the middle of the day, and they didn't want to be out in that. And they decided to go down into a gully nearby and just walk through it on their way back to the car before they were going to go home. And they were walking by and found an arm that belongs to a hominid. And then they found a piece of a skull. And then they found a femur. And they started to realize that what they had found was a skeleton, and they didn't have any repeats. So if you find a right arm and then you find another right arm, you have at least 2 people represented there, right? Because we only have one right arm. But in this case, they'd only found single copies of each. And they managed to trace it back into the part of the hillside where Lucy was eroding out of, and realized that they had this really fabulous skeleton. So they marked her, rushed back to camp to come say, oh my God, we found this amazing thing, and then proceeded to come back with the full team and start digging her out of the hillside there. And they were so happy and kind of elated to have found this fabulous, incredibly complete, but very tiny individual that they were listening to the Beatles' Lucy in the Sky with Diamonds over and over and over again. And that's where her name comes from. So they gave her Lucy from that song. And they were pretty sure that she was female because of her small stature. So she's just over 3 feet tall, and that the shape of her hips. So the nature of having to give birth means that women tend to have a larger inlet in their pelvis because that's got to be able to allow a baby to pass through it. And you can see that in Lucy's, that she's got that wider space. So they were pretty sure they'd found a female individual. And she has a bunch of things in her that were sort of a unique moment. So we'd found partial skeletons in hominin fossil record before, but they were much less complete. So there's STS-14, which is in South Africa, amongst Africanus. But that one was pretty small. It's mostly some vertebra and a bit of a pelvis. Lucy, she's got pieces of her head. She's got body. She's got limbs attached. And so she became this fabulous moment of here's like the whole picture connected together. And so if we had a kind of an ongoing discussion in the field at the time of, so we know that humans have really big brains, and we know that one of the defining features of us is walking on 2 legs, and sort of trying to figure out what came first. Did we get bigger brains and then we needed to use tools and then we stood up, sort of was one option, or did we start walking around and maybe that gave us the opportunity to get to energy rich foods and get big brains? And so this was an ongoing discussion, and there were sort of no good ways of knowing that. And Lucy became one of the ways that we could see that, because she has not a complete skull, but enough of a skull that she has a really tiny brain. Her brain is just a little bit bigger than what we see in chimpanzees, but the rest of her anatomy, so her hips are very, very human-like. So if you think about the way that our pelvises are, humans have ilia, the sort of flared part of our pelvis, that sit out to the side. And that's really important because it puts some of these muscles, or that are called our lesser gluteals, on the outside of our legs. And so when we stand up and we stand on one leg, gravity would like to tip the pelvis down to the side that's not touching the ground, that we have our leg lifted up for. And those muscles work to keep our pelvises straight on. And it's really, really important to do that in order to walk upright on 2 legs without wasting lots of energy wobbling side to side. So if you ever see something like a chimp stand up, the way their hips and their muscles are, they're actually aligned to the back, which makes them really powerful climbing muscles, but doesn't help them when they stand upright. So that's why chimps toddle back and forth and waste a lot of energy. So if you want to be a good biped, that's one of the critical anatomies that has to be present. So Lucy's got that. She's also got not quite enough remnants of her skull, but we can see then some later skulls that help show that the foramen magnum, which is the big hole at the base of our skull where our spinal cord goes in, on a biped, it's got to be right underneath the skull because we're upright and it sort of sticks our head on top of our spinal cord, over our hips, over our feet, keeps us nice and upright. Whereas if I'm a quadruped, that hole is at the back of my skull so that my head can be kind of facing out where I need to and my spine comes out the back. So Lucy's got that towards the bottom of her skull. And that she's also got a spine that shows a lot of the really important shape changes that are necessary for being a biped. So our spines are S curves. So we have lordotic curves in our neck and in the sort of lumbar lower back region, and then kyphotic, so the sort of convex, yeah, there we go, ones in our thorax and lower spine. So we have this kind of S going on. And what that does is it sort of serves as a spring as we're standing upright, and it also helps keep our head right over our hips. So sort of keeps our whole center of mass right down the midline. Right. Whereas most animals are either fairly straight or they have kind of a C-shaped spine. So even kids, babies, humans are born with a more C-shaped spine. And as they start to stand up, they end up getting that S-shaped. And we have Lucy's vertebra, and we can see from her lower spine that she has this lumbar curve. So she's standing upright over her own hips. And as I mentioned, the thing that made me want to go do this, so if we look at her ankle, which we don't have a lot of her feet, but we do have her talus and we have the bottom of her tibia. And then we can see she has that nice square bottom to her tibia saying that she's putting forces pretty evenly throughout her foot. She isn't primarily climbing all of the time. So she has these really cool suite of features, and she became this really cool, I have some mandibles over here, and I have a bit of a knee over here. How do I know who they belong to? She became sort of the gold standard of, who do I compare you to, and do you match up with her? Are you from that same species? And it sort of became that sort of very important comparison for a lot of other fossils that we found after her.
And I believe that the femurs slope inwards as well, something like ours.
Yeah, so we can see her femur has a bit of a crushed knee. So reconstruction of it absolutely has a bicondylar angle, but then we have found other afarensis that absolutely have the bicondylar angle, which is the really important. So when our femurs are actually sort of sloped in, and it puts our knees right under our hips, and it's doing the same thing as keeping us in a nice straight line, so that when we stand, we're really efficient and we don't
center of gravity, yeah. Lucy was the first example of Australopithecus afarensis, have we said. But what about other afarensis fossils that have been found since 1974? There have been males, females, infants. What do all these specimens tell us about this species?
Yeah, there's been an amazing number of afarensis discovered. Actually, afarensis is one of those species we actually know quite a lot about. And so sort of the most fabulous kind of next discovery right out of Hadar, which is where we found Lucy as well in the Afar region. The next year, Don Johansson and the team went back again, and they found the 333 site, and they actually found 13 other individuals from Lucy's species. And so these included adults, these included kids. And so what that site did was give us this huge group of folks covering all kinds of age ranges, because how we grow changes how we look too. So if you're going to know, is this a different species? Is it just a young version of something? You need a nice kind of time set that shows that. And so that's what the 333 site did, sort of, and it proved some of the things that were already sort of pretty sure from Lucy as well. Things like the location of the foramen magnum showed that there's significant sexual size dimorphism in afarensis. So one of the things about Lucy, she's just incredibly tiny. She's just over 3 feet tall, and so it's sort of odd to imagine that this is an adult of another species kind of walking around. She has fully erupted wisdom teeth, but they're not very worn. So she's an adult, but she's still relatively young. And that we see that there seems to be some individuals in this species that are as tall as almost 5 feet. So what we think is that their males and females are very different in size. So modern humans today have very low sexual size dimorphism. So really, there actually isn't all that much difference between how males and females look and how they're sort of organized. But if you think of something like gorilla, female gorillas can be considerably smaller than male, silverback gorillas can be almost 400 pounds and very, very tall. They have giant canines, whereas that's not the case for the females. So there are a lot of different primate species even that have a lot more sexual size dimorphism than we do. And that seems to be true in afarensis as well. We also know a lot about their feet. So we have practically every element of their foot has been recovered between the 333 site and several others. So we found the year before Lucy, Don Johansson actually found a femur with a really beautifully preserved bicondylar angle. And after these sites had been discovered, realized that also belonged to an afarensis. And that there have also been afarensis not only found in Ethiopia, but also in other sites in Eastern Africa, including at Laetoli, which is where my paper is about. And so that the type specimen. So when we look at a species, we want to say, how do I know that this particular individual belongs to the species? We sort of set what's called a type specimen. So this is the comparison sort of, this is our great example. Everything else kind of has to go with this or within the sort of range of things around that. Right. And so the type specimen for afarensis is actually from Laetoli. It's a Laetoli hominid number 4. And so they also show that the feet are incredibly human-like. So afarensis has a big heel. It's a big kind of puffy, so it's well made to hit the ground and to take a lot of that force that we have when we walk and have that sort of nice heel strike. They have toes that are pretty much in line with their feet, and then the big toe has a little more, it's a little more abducted, it sticks out a little bit more than what we see in a modern human, but nothing nearly so much as what we see in the other living primates like chimpanzees, where it's a grasping foot. They have toes that are a little bit longer, a little bit curved, because they probably did climb up in trees, certainly for food and certainly to sleep, avoid predators, but that they're tilted upright, so they're pushing off with their toes as well, so that they are, in a lot of ways, their lower limbs very much look like what we see in modern humans today. We have found even infants. So one of the most beautiful and really well preserved partial skeletons is from a site called Dakika. And this is of a baby or juvenile afarensis. And it has an amazing hyoid that's been retained through the fossil record. Hyoids are so small and delicate bones. They're the ones that are in our neck kind of floating. They don't touch any other bones. They're really important for the way that we make sounds and be able to understand and reconstruct sort of the capabilities of the vocal tract in afarensis. And so the fact that we have one from a kid is really amazing for Dakika. And it sort of shows they probably couldn't do quite what humans can do in terms of their speech capabilities, but there are some anyway. So that's really cool. And then the other one, a little bit older than Lucy herself, and then you had another fabulous fossils called Katanumu. He's about 3.6 million years old. And so that was found in Woronzow Mile, which is another site in Ethiopia. And it's another partial skeleton. And he is about 5 feet tall. So he's significantly larger than Lucy herself, and a little bit older as well. So kind of showing that this has quite a long time that afarensis was hanging out, and that this species has quite a lot of diversity within it.
Your paper examines the famed Laetoli footprint trackways discovered in the 1970s in Tanzania. This is an amazing site with some of the oldest evidence of bipedalism in the human lineage. There are other trackways as well, but before we get into that, can you give us a story of the Laetoli footprints and how we know it was Lucy's species that made them?
Yes, so Laetoli, which is a site in Tanzania, is one of the coolest and also most important paleoanthropological discoveries ever made. And so the site for Laetoli is kind of a unique place, and it's sort of made possible, if you will, by the volcanic activity that was very present in Eastern Africa. And so volcano nearby erupted, exploded, threw down a bunch of this really fine ash that then got rained on, and that rain turned that ash into mud. And then, luckily, thousands of animal prints were created across it. So, we have ones from elephants and dik-diks, which are little antelopes and rabbits, and all kinds of animals walked across the surface of that mud. And then, luckily, relatively quickly, the volcano exploded again and threw down another ash layer, and that buried those footprints and sort of sandwiched them into this moment in time where if it hadn't buried them again, they'd have been dried out by the sun or washed by rains again, got stepped on by other things, and they would have been destroyed and they just crumbled apart. But because they got kind of sealed under that next ash layer, they're protected. And so this site is about 3.66 million years old. And the actual discovery of it is kind of funny. So folks have been working at Laetoli for some time. So this is where Mary Leakey and her team were. And in 1974, that's when they'd actually found Laetoli hominin number 4, which is the type specimen of afarensis I mentioned. But they were still working at the site and looking around. And so they'd come back to a new field season in 1976. A number of researchers working at the time, and they actually just kind of got bored one afternoon and decided, as one does out in the field with not much to do, to have a dung fight. So they started picking up elephant dung and frisbeeing it at one another. And one of the researchers essentially hit the deck to try and dodge this, and realized as he was looking down that he saw this layer where there were these very distinctive little drops. And he had seen a very similar thing before, and what they realized they were fossilized raindrops. So a drop of rain hits mud, it makes a little impression, in the spot. And so kind of went, hold up, please stop throwing dung at me, and started looking at that. And that sort of found the tuft layer where these fossil footprints were and began this excavation. So in 1976 is when the first bipedal trackway, so there were several bipedal trackways, were found. So that's Trackway A, which I will talk about more later when we talk about my paper. But then that was a set of 5 prints. They came back again 2 years later for a field season in 1978, and that is when they found Trackway G. And Trackway G is fabulous. It's a set of bipedal prints. There seem to be at least 3 individuals here. So we have a fairly small individual walking on the left, and next to them is a larger individual leaving prints on the right, and at least one, although there is some research that suggests maybe even 2, a third individual was walking in the footprints of the larger individual. And so there's a set of at least 54 prints that go beside one another and preserve this incredible snapshot of bipedal locomotion. And so we know that they belong to hominins because they have to have the same kinds of architecture in their body in terms of the anatomies of the hips and at the knee in order to sustain that kind of bipedal locomotion. They're not bouncing up and down from being on all fours to upright. They're just striding across the land, extended hips, extended knees, the way that we walk. That a lot of the ways about how their footprints works, we can actually kind of take a snapshot of sort of what the footprint itself looks like inside and how did their feet move through that. And we see that they are doing pretty much a nice firm heel strike the way that we do, and that they're pushing off their big toe. They're not quite as good as what we see in modern humans in terms of how effectively and efficiently they do that. A little bit more of an abducted big toe, a little more mobility there than what we see in ours. They don't seem to have quite as much of a medial longitudinal arch. And so our arches are one of the things that kind of makes us really elastic and really good at that push off. And it's not something that is present in some other primates like chimpanzees. So they have one, but it's not quite as developed as what we see. And they seem to match up incredibly nicely with the skeletal elements we have from afarensis. And we have found afarensis fossils at Laetoli. So the type specimen is there. There have been a number of hominins of multiple specimens, multiple individuals that have been recovered from Laetoli, and they all seem to belong to afarensis in terms of things of the same age as what we're seeing in terms of the footprints. And so they match anatomically with what we know about the foot of afarensis, and that time frame matches up really nicely. And so those are the fabulous ones from 1978, so that's Trackway G. And then in 2016, published on another trackway, that's Trackway S, that was found. That's a set of 2 individuals walking, and those also seem to belong to afarensis, though they do belong to individuals that are a little bit larger. So it's sort of another piece of evidence that there is significant size dimorphism happening in afarensis. And so they're incredibly cool, and footprints are sort of this unique moment because not only a fossil, sort of everything that lives that could become a fossil has to die in a circumstance where it could become a fossil and not be instantly destroyed by being crushed by the ground around it, have something step on it, or whatever, and then it has to be found. And that's a pretty hard thing for skeletal material to do and end up being a fossil we find. So sort of finding Lucy in that moment was incredibly rare. If they'd been there 5 years earlier, she'd have been buried in the hillside and they wouldn't have known. It came 5 years later, she might have washed away completely and you would never have found her again. So that you have to have the right moment. And then footprints are even rarer because the sort of moment of catching the snapshot where you walked but then get buried right away is very rare. So they're a fabulous, unique moment, and they're sort of unequivocal about how they moved, right? These guys are upright, walking on 2 legs, where sometimes we have fossil specimens. Certainly we have some stuff that's older than Lucy and her species, where we say, well, we're pretty sure based on, some of these anatomical pieces that we think these animals are walking upright, but footprints are, I got it. There's no doubt to them. They're walking upright on 2 legs. And so they're sort of one of the oldest unequivocal moments of upright walking.
I think you've got one behind you there, don't you?
I do. So this one is from the other site. This is from Laetoli A. So we'll come back around to it. But so that's 3D scanning. You can see how much they look like us. And so very much a cool moment to kind of imagine the American, the American Museum of Natural History has a really cool reconstruction of the potential of what the site of Laetoli G may have been. So they have the smaller individual, the two footprints tracks are really close to each other. So the smaller individual and the larger, they actually have them sort of painted as if they're walking with their arm around each other. And that's not something we can know, but it is absolutely something that kind of captures the imagination that this sort of family group is walking sort of through this landscape, which would have been a little uncomfortable given the volcanic explosion, but not so much that all the animals left, clearly, because they're all still hanging out and moving through this, probably moving towards a water source. Laetoli itself is a fairly dry place, so they're probably heading towards some kind of water.
Probably evacuating, and they had no choice but to walk across it, no matter how hot it was.
It's not running, so we're not sort of oh my God, the explosion's around us, but probably don't want to be here much right now. Interestingly, the smaller individual of Laetoli G seems to have something going on with their right leg. So the way that their footprint is sort of like they're limping or hurt. So kind of an interesting, maybe they're helping one another walk on. It's something that we can't know that for sure, whether they would, but it makes for a really cool image to imagine that kind of humanity, if you will, extends back that far in time.
Well, the afarensis footprints were just one of the trackways found back in the 70s. An earlier trackway was eventually attributed to bears, animals also capable of walking upright. But you and your colleagues have shown that this isn't exactly the case. Isn't that right?
Yes. So Laetoli A, that first trackway that was discovered, when they originally found it in the 1970s, it was weird. So this is an example of A, and it's got really wide feet relative to what they were seeing in the afarensis skeletal material. It just doesn't quite match up with any of the things they had found then. And sort of the prevailing theory at the time was that there was really only one hominin species existing. And that was partially due to the fact that our fossil record wasn't nearly so complete 40 years ago as it is now. And so it was like, okay, this doesn't seem to match up with the afarensis. And then when they found G 2 years later, which does beautifully match up with the anatomical evidence of afarensis, like, well, I have lots of these prints at G. They very clearly match up. I have a nice story here and I can understand this. And then A kind of fell out of study. We didn't know what to do with it. And sort of the best guess at the time, based on what was available, was that maybe it's a bear. And often people will hear bears in Africa and they kind of go, what? There were bears that lived in Africa about this time in Eastern Africa. These are called Agrotherium africanum. They're quite a large bear, sort of in the grizzly bear, polar bear sized kind of thing. And so this would have been a juvenile bear walking alone for 5 steps that would have had its footprints. And even in the 1970s, the researchers were sort of bear ? It was sort of an unsatisfying answer, but they didn't have a better suit for it. They didn't know, kind of a Cinderella moment. We have a slipper that doesn't seem to fit any feet that we know about. And so then it fell off the radar and folks really weren't thinking about it very much. And then when I got into my dissertation, because I was interested in bears as a really good analogy for apes, I started thinking about bears, looking into things about them. And that sort of research from the early 1970s showed up. And I kind of went, I don't think that it's a bear. And it sort of doesn't quite match up with what you might expect from that. And I was incredibly lucky. I was doing my dissertation at Dartmouth College. Just down the road from me in Lyme, New Hampshire, was one of the world's experts on American black bear. So Dr. Ben Killam runs the Killam's Bear Sanctuary for the American black bear. And so one of the things they do is take in orphaned black bears. And so there was a set of cubs about the size of the prints at Laetoli A. And I said, I'm going to have them walk through mud and see if they match up. And so I took a little trackway made of mud and applesauce and maple syrup, and I had little guys stand up and walk for me unassisted and sort of, what do bear footprints look like when they do that? And they don't look like this trackway. And part of the reason for that is the anatomy of bears. So as we've talked about, afarensis and Lucy has the set of anatomies in her hips and in her knees that are going to make her really nice, stable, upright biped. And those aren't present in bears. So when bears stand upright, they have the same thing happening where they're wobbling. And they can't have, one of the moments that makes these prints at A very unique is that there's a moment where they have cross-stepping. So if you think kind of the extreme version is a model walking down the catwalk, where you're walking and you're kind of crossing one foot all the way over in front of another. And that is obviously not something we do frequently, but that is something humans can do, clearly when they're model walking, or if you're playing around, or even on slippery circumstances, we might do that. But it's not something a bear can do. It's not something a chimp can do, because they simply can't maintain their balance on two legs and cross their feet like that. And so that was one of the first kind of moments of this doesn't seem to be possibly a bear. It's got to be a hominin. And so we started going back and looking at finding colleagues who had data on chimpanzees and looking how they walk. Sort of coming from that, we realized that the original prints in the 1970s hadn't been fully excavated. So our team actually went back to Laetoli and found these prints at A. And one of the things that it turns out, we couldn't find any casts of the originals. When they were discovered, they took casts that were really good, but they seem to have disappeared. And they may be somewhere in the world, but we couldn't find them. And so we were really afraid that actually the whole fossil trackway had been destroyed, because it didn't seem like they'd actually reburied the trackway after they had found it. So we were, 40 years of rain and sun will do a great deal of damage to something. So we were very lucky. We ended up using the maps that Mary Leakey and her team drew when they actually did the initial discovery of the site. They took these fabulous maps, not only of the bipedal trackways, but of the trackways of other animals around them. And it turns out there is an elephant trackway very close to Laetoli A. So when we went back, we actually managed to find that elephant trackway and use it to rediscover the A site. And those 40 years of rains had washed sediment on top of the Laetoli A, as opposed to destroying them. So they sort of essentially reburied them and kept them preserved just the way they had been for almost 4 million years before that. So we managed to find them again, excavate them, actually do the nice complete excavation. We managed to find some anatomies that hadn't been fully excavated before. So things like the impression for the second digit, just really important, because primates, including humans, have a very large big toe and then a relatively small set of seconds through fifths, whereas bears are actually the opposite. They have a big fifth and then relatively equal-sized other ones. So that was one of the reasons why bear had been suggested for these, is instead of having a cross-stepping moment, if I'm a bear and actually that big digit is the fifth, I sort of solve that issue. And then it turned out that those proportions don't match. They're actually much more similar to the large size disparity we find in primates than what we would find in a bear. So really clear now that that does have cross-stepping. And if you have cross-stepping, you're a hominin. It's the only way that that works. And so we know not a bear for sure. And then the next logical question is, we know that there are afarensis prints there. Are the Laetoli A just more afarensis, which have been fabulous? And so we went and compared the prints to G and to S, and it turns out they actually don't match. So the proportions of Laetoli A, they're actually much wider than what we see in terms of the S prints. They also happen to have sort of different internal morphology. So if you compare any two human prints, which is, we had two really well preserved A's, any two human prints to the average human, you can get a nice distribution of how different within a population they might be. And you can do the same thing with any two chimps. And so we have another group here. And so there's a little overlap between humans and chimps, but not a lot. If we do the same kind of thing looking at the S prints and the G, they actually fall within the human distribution. They aren't all that different from one another and from human prints. And if we do that same analysis with the A prints, they are as different from a human as a chimpanzee foot is. So they are not chimps. They don't look like a chimp, but they're that different. They have very different proportions going on. They also don't seem to match in the way we might expect. Well, maybe they're just a young afarensis because they're quite tiny. And we do change over time. So we looked at how humans who are young versus old, whether they're modern humans today, or we looked at a fossil footprint site called Ngaro Cerro, which is about 19,000 years old and has thousands of modern human footprints in it of all ages, and sort of how, folks in the past walking in very similar circumstances. They're walking in volcanic ash. Folks living today, chimpanzees, and we can see kind of the growth trajectory within those groups. And here are the G and S prints. And, is A just on that line? He's just different because it's young. And it turns out, no, it doesn't match that. It's actually much closer to the line of progression what we see in chimps. So this is an animal that is moving differently from what we see in the other two Laetoli bipedal trackways. And so somewhat unsatisfyingly, people want to know, who is it? And it's like, I don't know yet. We don't have a good set of, we're still kind of missing our perfect shoe to slipper moment for Cinderella, but we do know that the foot of afarensis doesn't match. So what it's cool, Laetoli A says that we had two hominins living at exactly the same time, which is very hard to find in our fossil record. So before we have sites both in Eastern and in Southern Africa where, big time ranges, we have fossils that we found here, and this sort of our time ranges have a little bit of overlap, but that overlap may be tens of thousands of years. So they were in the same place, but were they there at the same time? It's hard to tell. Whereas at Laetoli, we know they happened at exactly the same time because footprints are made in sort of scales of hours to days. So the Laetoli A individual could have looked up across the landscape and seen the afarensis walking around making the G prints, which is a pretty fabulous and unique moment in our fossil record. So it's been a really cool joy to kind of work on that site and kind of catch this moment of science where we kind of come back around knowing new things, having new technology, having the benefit of a fossil record that's changed significantly. And we know there's a lot more species and a lot more diversity in how those species were moving through the world, and kind of looking at the fossils we had examined before with fresh eyes and kind of seeing new things. And that's how science works. It's a collaborative effort, kind of using the best information you have, and then when you have new information or new ways of getting at that, kind of making the next step forward. And that will happen again with our stuff by somebody else, which I love.
And I suppose one day someone will discover this hominid and match it to the Trackway A footprints.
Yes, which would be absolutely cool. There's a couple sort of candidates, but it's one of those moments we have a fabulous cranial dental material from Neanderthal or Australopithecus dyremida. There's sort of options. We don't have their feet, so we don't know what they look like. And we have cool feet like the Laetoli foot, but unfortunately that's also from Laetoli in Ethiopia. But we don't know who they are.
Well, I guess Lucy will always be the poster child for human evolution, and no doubt the fascination with her will go on. So what's next for you, Ellie? What future projects can you tell us about?
Yeah, I have a couple things that I'm working on. They'll be a little while yet before they come out. A coworker and I are looking into some of the anatomy and sort of within the bear family and kind of using them as a model. There are bears who are terrestrial. There are bears that are, very arboreal up in the trees, and kind of looking at how their calcanei changed and sort of use that to help us kind of interpret and understand our own fossil record for hominins. So that's sort of a project that's in process. And then I also am working on using micro CT and being able to look at how bone, particularly our calcanei, how it's affected by our foot posture. So are you plantigrade or some degree of digitigrade? There's semi-plantigrade. There's sort of a spectrum of foot postures there, and how those change the sort of mechanics inside the bones themselves, and try and understand that so that we can use that to apply. Our fossil record is amazing and lovely and frustrating, particularly for folks like me who study sometimes some pieces of feet, because you may not end up with a full bone. So can we find ways to sort of interpret some of the internal morphology of bones and kind of get a snapshot from them, even if you don't end up having the full specimen represented? So that's sort of another angle that I'm currently working on. Those are the two big ones.
I will leave links to your social media and research papers in the description below. And all that's left to say is thank you once again, Ellie, for coming on to Evolution Soup.
Thank you very much for having me. It's been lovely chatting with you.